Hello, my name is Mike Raj, and this presentation is a short presentation. It's about glycolysis, gluconeogenesis, and the role of aldolase gene expression. And this is for concepts in biochemistry class. It's a very basic introductory presentation into the different cellular mechanisms involved. So let's begin. So these are the cellular mechanisms that I'll be presenting: glycolysis, gluconeogenesis, and aldolase, which is an enzyme. So what is glycolysis? Glycolysis, um, glycolysis pathway. Glycolysis is the breakdown of glucose. Glucose is a six-carbon carbohydrate molecule which is required for the production of ATP, which is adenine triphosphate, which is the basic cellular currency necessary to run the human body and for most cellular mechanisms in other organisms. So the first step of enzymatic degradation and the breaking of glucose to pyruvate. It's a long chain process. Glucose is six carbon, while pyruvate is three carbon. So there are mechanisms involved to slice in systematic fashion. So pyruvate can ultimately be turned to ATP, adenosine triphosphate, which is necessary for the cellular mechanisms to function. If there's a low shortage of ATP, especially for smooth muscles like heart, brain, and similar muscles, these muscles will quickly weaken and start to die because they rely on ATP to function, especially heart. During glycolysis, the end product can be pyruvate or uh, depending upon the process of breakdown and what enzymes are functioning. Another name for glycolysis is Embden-Meyerhof pathway. And glycolysis does not require oxygen, just like citric acid. It does not require oxygen, and it's done within the cytoplasm. While citric cycle is done in the mitochondria. So for one glucose molecule, six carbon molecule, it can be split to two pyruvates, which pyruvate is a three carbon molecule. So here you can see from glucose, the enzyme glucophosphate isomerase turns glucose to fructose 6-phosphate, and phosphofructokinase turns this molecule into fructose 1,6-bisphosphate. And you can see here you can see aldolase. This is a vital enzyme which can convert this molecule to dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. It can interchange, and likewise aldolase can also change these two molecules back again to fructose 1,6-bisphosphate. And this we look at this cycle here, and triose phosphate isomerase enzyme changes this molecule to glyceraldehyde 3-phosphate. And here you can see ATP turn to ADP. So um, certain extraction or conversions require energy, while certain extraction or conversions tame energy. Here ATP is changed to ADP, which is use of energy, and here NAD plus is changed to NADH, which is conservation of energy. And from here to glyceraldehyde 3-phosphate dehydrogenase turns to 1,3-bis phosphoglycerate. And again, and here energy is used. No, energy is conserved where ADP is changed to ATP. And phosphoglycerate kinase changes to 3-phosphoglycerate, and phosphoglycerate mutase changes it to 2-phosphoglycerate, And here the water is removed and enolase. So here is enol pyruvate, PEP also called, and the pyruvate kinase ultimately changes conservation of energy here, ADP to ATP, and finally changes it to pyruvate. Pyruvate goes into the citric cycle, and then ultimately, which happens in the mitochondria, then ultimately it gets converted to ATP. So gluconeogenesis pathway. So what is gluconeogenesis? Gluconeogenesis is the formation of glucose from pyruvate, mainly from non-carbohydrate sources like fats, lipids, and so forth. Stored fats and lipids mainly from the liver. This happens when one starts fasting, not intermittent fasting like 1 or 2 days, but in a prolonged fast, it really kicks in. Being an intensive fashion after a week or so, then gluconeogenesis takes in because people usually have fatty liver. It stores up as a form of fat, and in the muscles is stored up as the glycogen. So mainly occurs in the liver, constantly feeds other organs if there's a need comes because of fasting or if the body is in an intensive process of exercise and so forth, if you go to gym and so forth. So somewhat similar to glycolysis, but not the exact process. So many of the same enzymes that function in glycolysis also function in gluconeogenesis pathway. So here you can see glycolysis and gluconeogenesis. Here you can see aldolase, the same enzyme of concern, still working here, changing these two forms into fructose 1,6 bisphosphate or transferring ultimately to glyceraldehyde 3-phosphate. So glycolysis takes glucose and breaks it in a catabolic process, breaks into pyruvate right here, while gluconeogenesis takes up the pyruvate and builds it up. As you can see, it goes up there to make it glucose, and ultimately will be uh, stored in as a glycogen for quick energy release. And this is from carbohydrate sources, and this is from non-carbohydrate sources. So So this is the enzyme of concern, aldolase. Aldolase is a glycolytic enzyme that catalyzes the conversion of fructose 1,6 diphosphate to glyceraldehyde 3-phosphate and dihydroxyacetone phosphate via the glycolysis metabolic pathway. So this is the enzyme that helps convert this molecule to either molecules. This it has that flexibility with it, and it can go either up, break it down, or build up. So enzyme involves in reversible conversion of, as I mentioned, fructose 1,6 bisphosphate to glyceraldehyde 3-phosphate G3P and dihydroxyacetone phosphate, just DHAP. So we come to aldolase isoforms and expression patterns. So isoform is a different forms of the same enzyme, and expression patterns are termed of how the gene is expressed in a particular fashion. So ALDO gene is the gene that actually codes for aldolase and its different isoforms, and is mainly found in human chromosome 16, though there are certain pseudogenes found in chromosome 3 and 10. So there are three isoforms which exist: A, B, and C. all three forms are found in the human body, but mostly it's A that is expressed. Aldolase A is mainly expressed in the liver and is the most ubiquitous gene that is expressed throughout the body. Every cell uses aldolase A expression, though overexpression results in cancerous growth, like lung squamous cell carcinoma (LSCC), renal cancer, and hepatocellular carcinoma. Well, why is that overexpression of this particular aldo gene contributes? It doesn't directly contribute to cancer, but it does enhance or mm, reinforces the cancer cell and helps it to spread. So why is it? So overexpression of this particular gene results in excess glycolysis, which helps in metastasis of the cell, meaning it becomes more cancerous and spreads easily because it has more vitality, as aldolase helps to break down and release um, help help to in the process of breaking down glucose and or in the transference of fat uh, to glucose and ultimately produce ATP for the benefit of the cancer cell. Because cancer cells don't necessarily have to go through the entire process to get the energy to get the ATP. It can use the subverting mechanism of using lactic acid and so forth to get energy derivation. As a cancer cell requires a tremendous amount of energy and ATP, glucose to function. So it constantly needs more and more and more to function because as the normal cellular functioning as like in the glycolysis or gluconeogenesis process, it's not entirely required. It but this still requires aldolase isoforms because it's like an intermittent enzyme that helps to either break it down or build it up, and it helps to speedy release of ATP into the into the cell for function. So, what is the transcription factors? Transcription factors are proteins that control the transcription of the genetic code. In this case, it is of aldolase enzyme from DNA to mRNA. So transcription is the process of uh, taking a gene which is in DNA and transcribing into an mRNA. That is called transcription process. Where translation process is taking that mRNA codon and converting into a protein through a ribosome. Uh, so that's why transcription factors are important. So one note here, TF is necessary for proper expression of genes. Genes could be normal, they could be normal and good and have no defects whatsoever, biological defect whatsoever. But if transcription factors fail, then the person will experience like diseases and uh, as if they did not have that reliable gene set, or if their genes were dysfunctional or miscoded or corrupted in any way or shape or form. So transcription factors is quite necessary. Conclusion. So what is for conclusion for this short presentation. So glycolysis is the process of breaking down glucose to pyruvate, while for glucogenesis is buildup of glucose from pyruvate acid or pyruvate. And aldolase is an enzyme necessary for both pathways, and overexpression is one of the signs of cancerous growth. And these are the references. This is the textbook. So once again, thank you for listening to my presentation. It was about glycolysis, gluconeogenesis, and the role of aldolase. Gene expression. Was a quick presentation, simple presentation on these subjects, and this is for concept in biochemistry class. And my name is Mike Jensen Raj, and thank you once again for listening in. Thank you.